as Jay said, Yvonne and I are from Team Tox. That stands for Trainees of Environmental and Molecular Toxicology. We are actually both PhD students in the toxicology department at Oregon State, and we are the outreach chairs for our organization. So once COVID shut things down and we were looking at remote learning, we were trying to think of ways that we could still engage with the community and continue some of our outreach activities, which usually involve working with students anywhere from elementary school up through even undergrad programs. So we started to think about taking some of the activities that we already had and transitioning to remote platforms. And that's sort of how this activity that we'll go over today was born. So to give you an overview of what we'll go over today, we'll be talking about why we created the activity, and we'll give you an overview of the Radish dose response activity and what it looks like, how the students sort of go through it, and the different resources that we've developed for the activity, as well as some of the main learning objectives that we'd like students to take away from the activity. So hopefully we can get you excited about it, and you can ask any questions that you have during the workshop. Okay, so when we decided to choose the Radish seed activity, our larger purpose was to teach students about toxicology and environmental health, which are largely the focuses that Yvonne and I have in our program. And talking with students about just different chemicals that we're exposed to on a day-to-day basis, and thinking about ways in which we can test those chemicals to know if they are toxic to us, and then taking those concepts and applying them to larger systems. So how does this impact our environmental health, and how do humans and ecosystems interact with each other on a larger scale? Some of the more specific objectives that we were hoping to hit with this activity, as far as concepts, were different kinds of exposures to different types of chemicals that you might be exposed to, the concept of dose, or how much of a chemical you're exposed to, and relevance to our everyday lives. And then in addition to that, we wanted to touch on concepts that had to do with experimental design, like writing a hypothesis, identifying independent, dependent, and control variables, asking the question of what is replication, and sources of error in an experiment. And then after the activity, we have some discussion questions and activities for plotting and analyzing the data that the students come up with. And those activities and worksheets we'll show you a little bit later, but those do differ based on the level. So for middle school, their data analysis is a little simpler compared to the high school level. But both levels hopefully engage some critical thinking and discussion. So along with the activity itself, which we'll go over, we created a series of videos where we were introducing environmental health and toxicology. So that's this video that I have a screenshot of, and I will show this to you. We also created videos for each day of the experiment, sort of walking the kids through how to set it up. So with that, I will show you this video as an example.
Hello, everyone. We are students from the Trainees of Environmental and Molecular Toxicology organization at Oregon State University, also known as Team Tox. During this video, we will talk about environmental health and toxicology and why they are important. We can think about environmental health from two perspectives. One, Think about human health and safety. And 2, thinking about the health of the environment, plants, animals, ecosystems, and more. For now, let's start talking about human health. Humans around the world are exposed to factors that can affect their health, like air pollution, smoke, metals, chemicals, and other things. For example, we might be exposed to things on our drive to work or school, at our jobs, or even in our homes. Some chemicals, like soap, are really helpful and are not dangerous to us when we use them as we are supposed to. But some chemicals can be harmful to us or the environment, and we either need to be careful to limit our exposure to them or to wear protection when working with them. This idea can be summed up in a concept called environmental health. It is the science and practice of preventing human injury and illness and promoting well-being by finding, evaluating, and limiting exposures to hazardous things in the air, water, soil, food, and everything around us. Just like chemicals can be harmful to humans, they can also be harmful to plants, animals, and the environment. For example, if some chemicals are dumped into the water system, they can hurt fish and other aquatic organisms. But how do we know which chemicals and substances are harmful to us and other organisms? The answer is toxicology. Toxicology is the study of the harmful effects that chemicals and other substances we might be exposed to can have on living organisms like you, me, plants, or animals. Some chemicals can occur naturally in the environment, like a chemical called urushiol, which is produced by poison ivy, or may be processed and manufactured by humans, like pesticides we use to keep pests from eating our crops or the gasoline we put in our cars. A key concept in toxicology is that the harmful effects that a chemical can have depends on the amount of the chemical that an organism is exposed to. The amount of a chemical or substance that an organism is exposed to is called the dose. You may have heard the saying that the dose makes the poison. For example, if someone were to brush up against one leaf of poison ivy, they would likely be exposed to a smaller dose of urushiol than someone who is walking through a forest covered with poison ivy, brushing up against countless leaves. We can also use asbestos as an example. Asbestos is a fibrous material that is heat resistant and has been historically used in building materials, car brake pads, and more. It's very dangerous when inhaled and can cause cancer and other diseases. The doses of asbestos that people may be exposed to can be very different. For example, a person who drove a car with brake pads that contained asbestos would likely not be exposed to as high a dose as a mechanic who works on cars or a factory worker that produced those brake pads. You can find more information about asbestos at these video links. The effect that a chemical or substance has on an organism is called the response. For example, after being exposed to poison ivy, our response may be an itchy rash on our skin. To investigate how harmful a substance is and to compare the toxicity of numerous substances, toxicologists use a tool called a dose-response curve. This involves graphing the response you observe in an organism or group of organisms after exposure to certain doses of a chemical. A typical dose-response curve shows that as the concentration of a substance increases, the percentage of people or organisms that will experience an effect also increases. Toxicologists use model organisms to help them investigate the safety of chemicals or other substances for humans and other animals in the environment. Some examples of model organisms include worms, fruit flies, zebrafish, and mice, or even single-celled organisms like yeast and bacteria. When using model organisms, it's important to think about the similarities and differences between organisms. Think about what humans have in common with each of the organisms and what they could tell us about ourselves. What about differences? What are the limitations to drawing conclusions about humans from tests using other organisms, say a bacteria? Today we learned about two big concepts. The first was environmental health, what it was and why it's important. The second was toxicology. what it was, and how toxicologists use dose response curves and model organisms to investigate the safety of chemicals and other substances. So the next time you're out in the woods and come across poison ivy, or the next time you're using a chemical around your yard or house, you can think about the environmental health and toxicology.
So hopefully that gives you an idea of what our videos look like. So that was kind of the background information. And then, like I said, we had a few videos going over how the actual experiment setup went. And so in addition to the videos, we also put together some lesson plans for teachers. We put together some worksheets for the kids to follow along with, both while they're doing the activity and with some discussion questions and an opportunity to plot their data, which we'll show later. Okay. So with that, I'm going to pass it on to Yvonne, and she can talk a little more about the activity itself.
Thanks, Emily. So like Emily mentioned, we have a couple other videos that actually run through step-by-step how students can actually go through these activities. We were wanting these to be just extra support since we're not there in person to do the activities with them, just asynchronous videos that they can go to for additional information they might need. So a lot of these slides I've actually pulled just from the slides we used for the videos. So if you go back and watch them after the fact, they might look familiar. But the materials that the students need are relatively simple things to acquire, I think, so that's really nice. I think the hardest thing to acquire might just be the radish seeds. All you need is some plastic resealable Ziploc bags, paper towels, the radish seeds. The way we set up our activity, we had instant coffee as the substance that we included in the kits for the students to test. And then a teaspoon or measuring device, water, and then the associated worksheet. Thank you. So we asked the students to gather all of their substances together. And so that entails preparing the instant coffee that we provided in the kits. And then we also have the students select a chemical bee of their choice from around their house. So this way they can choose something that they're really interested about investigating the toxicity of, and then they can include it in their experiment, which adds a fun element. And we do provide the guidance that this should be a substance that's meant for human consumption, just for safety purposes, something that's meant to be applied to your skin or ingested, something like soda pop or hand sanitizer or dish soap, things like this. But they can really get creative. The next step is just to prepare the actual bags and treatments. So 2 paper towels get folded up and go into each sandwich bag, and the appropriate labels get put onto them to match up to all of the treatments that they'll be making. And then we go through the process of walking them through how to actually make their chemical treatments. And this will entail 50%, 75%, and 100% coffee solution treatments. So we provide them with this table, both in the worksheet and in the videos, so that they can follow the appropriate dilutions to get to the percentages that we want to test. And then for their chemical B, they can either choose to use the same percentages, the 50%, 75, and 100%, or if they want to, we also provide them with some additional equations that they can use to determine additional percentages if they want to change things up. And so then once they have all of their baggies with all of their different treatments all labeled, the next step is to actually put the radish seeds in. So 10 radish seeds go into each bag, spaced apart so that they have room to grow. And then the activity lasts for a duration of 3 days. And by the end of those 3 days, most, if not all, of the control seeds only treated with water should be germinated. And the other treatments will likely be a range between very few radish seeds germinating, potentially to all the way to the same as controls, depending on how toxic that substance is to the radish seed. So over the course of those 3 days, students will be collecting data on day 1, day 2, and also day 3, counting how many radish seeds are germinated versus not germinated. And then on the 3rd day, we have students calculate the percentage of the seeds that were germinated. And then that data goes into their plots that they will make. And like Emily mentioned, we have slightly different tracks for middle school level versus high school level activities that we set up for this overarching radish dose response activity. And so far, I've been running through what we set up for the high school students. And after the next couple slides, I'll walk through exactly what the differences are between those two activities. But like Emily mentioned, one of those is for the middle schoolers. They will create a bar graph with their data. Whereas with the high schoolers, as you can see here on this slide, we will actually generate a dose response curve. So in the videos, we step them through how to appropriately set up a plot in general with the right axes labels and making sure you have a legend and all of those key elements for a graph. And then the actual process of plotting their data onto this graph and then making this typical S-shaped curve that dose response curves usually follow. And another really exciting part of this activity is one of our outreach committee members actually created an app online, a web tool that you can visit and then go to in this left-hand side of the web page. You can put your data in that you collected during the experiment, and then it will spit out on the bottom right-hand side computer-generated dose response curves. So this can be used, and I know it's pretty small here, but we can include the actual link to this in the chat so that it's easier for you to access. But this is a great way for students to not only double check their plotting to make sure that they plotted everything correctly, but it also makes it a lot more accessible for students to just put their data in, get the graphs, screenshot it, and then share with each other, and then generate classroom discussions where everyone can share their data and compare. So jumping into exactly what's different between the middle school and the high school levels that we set up. So the students begin by dosing their radish seeds with different substances. And as you saw, what I presented from the videos, we asked the high schoolers to test 2 substances, and that's coffee and some chosen household products. Whereas for the middle schoolers, they will be testing 3 substances. We provided in the kits coffee, salt, and then they also get to choose a household product. And the difference here is mostly because of the next step in the actual creation of the treatments. The high schoolers are making treatments for different concentrations plus a control, whereas for the middle schoolers, we ask them to only test different substances at 1 concentration and then compare that to a control. And this was driven by our goal to focus on just slightly different objectives for each of these levels. For the middle schoolers, we wanted to focus a little bit more on understanding of experimental design concepts. So really focusing in on how we create a controlled experiment and how, when we do that well, we can generate good data. Whereas on the high school level, we wanted to focus a little bit more on slightly more critical questions of comparing substances across concentration ranges, and then also including a bit more discussion about implications of our findings from this experiment or other toxicology experiments for humans and environmental health.
I just have a quick question about just sort of logistics of somebody setting this up, and about how, thinking about once you get the experiment set up and going through that process, how long does it take for the seeds to germinate, and what sort of results are we looking at? There was one question in the chat that talked about, potentially eating the radishes, and I know we're not getting to radishes, but eating the germinated product, and definitely advise against that. Shouldn't eat experiments, but thinking about how long does this take, and what's the data that's being collected specifically?
Yeah, so those are good questions, and I would second that. If you're running an experiment, it's always good to teach good laboratory practices. So definitely don't eat the radishes, especially because it's really easy to mix up what your treatments might be. So in terms of the amount of time that it takes, the initial setup, I think, might take, and Emily, you can jump in if you remember this more clearly than me. The initial setup is pretty simple. I think it might have taken me an hour because I was multitasking. And then the actual collection of the data each day takes maybe 5 to 10 minutes. Really easy, just quick evaluation of the seeds. And then on the third day, just a little bit extra time for maybe doing a little bit of math with calculating those percentages. And then the bulk of the time can be spent on actually creating the graphs and thinking about what the data means, and then discussion afterwards.
I would also add, so we'll go over what the actual questions and graphs in the worksheet look like. So hopefully that gives you a better idea of what the data looks like. But as far as like what the setup involves, it's really like getting the seeds into the baggies, putting water in the baggies, and then whatever your treatment is. I think it probably only took me like, I don't know, 30 minutes, and we were trying to figure out how exactly the activity worked. But then on the consequent days, you're really just checking the seeds and saying either like, yes, it has sprouted, or no, it hasn't sprouted. So you're looking at your 10 seeds and saying like yes or no. So hopefully that is a little helpful. Like I said, we'll go over what the data input looks like. But if you have other questions, let us know. So I'll just finish up our spiel really fast. I just have some more details on our resources that we came up with, so you can see some more of the differences between the high school and middle school resources, so that we can discuss those a little bit as far as being age appropriate, and some of the NGS standards that we tried to link to the activities. So the first part of the worksheet is all about setup. It's about identifying your independent variable, your dependent variable, control variables, replicates, and then encouraging just taking notes, writing down a hypothesis and any observations that you see while you're actually setting up and performing the experiment. Yvonne showed a picture of the table where you can record the percent that were germinated each day. I don't have that included, but it is a part of the worksheet. And then there's the portion of the worksheet that has a plot, and you can kind of see for the middle school, there's a lot more information provided with the title and axes labeled. So it should be pretty straightforward for them to plot their data. And in the case of the middle schoolers, it's actually just a bar chart because they're comparing the same concentration of different substances. So they can just plot their bars for the number of seeds that were germinated on day 3. And they can compare then which chemicals were more toxic than other chemicals compared to their control condition, which is just water. And then for the high school, it's a little bit more bare bones. We trust that they can plot a graph at this point. So there's no axis labels, that is up to them. And then given information on how to construct a dose response curve like we talked about, they can plot their results for each of the doses and hopefully get a nice curve. And then at the end of the worksheet, we just have some discussion questions that we also incorporate in the lesson plan to wrap up the experiment and talk about what they did, as well as generate some conversation about larger applications. And this is sort of where the links to the standards come in as well, talking about not just these radish seeds, but if we're looking at toxicity of chemicals, how does that apply to like our larger population and human health? And how does this relate to pollution in the environment? So asking some of those bigger questions as well. For middle school, the discussion questions on the worksheet are pretty simple. Again, we were really focusing on experimental design. And so in addition to asking about the purpose of the experiment and why we need control variables, we're asking them to interpret their data, think about what they might do in a future experiment, and how they might reduce error in their experiment in the future. For the high schoolers, they're a little bit more complex questions. I won't go through all of these, but basically just going through the experiment with a little more detail and a little more critical thinking. So as far as linking to the NGS standards, we got a lot of help with Jay in thinking about this. We realized that it's important for teachers to meet these standards in their classroom, and they can't just throw in random activities. We recognize that there are objectives that need to be met in the classroom. So we really wanted to provide an activity that would be genuinely useful to teachers, and hopefully we have accomplished that. But we did try really hard to incorporate some of the standards. And so this is all a little new to me. So it was really helpful to have Jay, but these are some examples of performance expectations that link up with the experiment for middle school. So one could be constructing a scientific explanation based on evidence for how environmental and genetic factors influence the growth of organisms. So you could have a discussion in your classroom about what each of these substances are really doing to the seeds. Why are some germinating while others aren't? So you could talk about what salt really does to the seedlings. You could talk about why are some of the seeds in the same bag sprouting while others aren't. So that could be because of genetic factors. And then for high school, this is still in development. I was just talking to Jay about this last night, but there are some performance expectations that relate to mathematical representations and modeling for populations and ecosystems. And so this activity could be linked to that if you chose to have a discussion relating to those. So that is sort of everything about our activity. With that, we really wanted to talk to all of you and discuss the logistics of implementing this activity, which we've started to do some, which is awesome. If you have any comments for us, we would love to hear them. I have some general questions here to maybe guide discussion. And yeah, we would love to hear your thoughts. So I am going to mute myself for a second so other people can speak.
So one quick question I had is when when students are doing that data observation, so they got they fixed their toxicants, they've made their dilutions, and they've set the experiment up. Now they're looking at collecting that data. How do they determine? I assume they're looking for just germination, correct? And how do they determine that germination has occurred? What is the standard of which they're looking for? How do they determine that? Is that something they have to come to an agreement prior to the experiment happening, or because ideally you maybe in a classroom you're relating these across different students to gain more replicants? What are your thoughts on that?
That's a great question. And I definitely had the same question when we were going through this activity for the first time. I was like, I am writing down things that have germinated when I can, see the sprout popping out of the shell. And I forget, honestly, in the lesson plan or the worksheet, if we defined what was considered a sprout, but it would definitely be useful if the class could come to a consensus. Maybe we could provide like an image of what would be, germinated, because it is really hard to tell sometimes and a little subjective. Yvonne, do you have any comments on that?
Yeah, I know in the videos that we created, we have some pictures of germinated seeds, and the way we defined it was just essentially whenever you can see kind of that white new shoot coming out. I know that's defined just by some example pictures in the videos. I'm just looking through the lesson plans and not sure if we defined it there, but that would be a good addition for us to add. Or else just discussing as a class before everyone starts data collection. That's a good point. Thank you, Jay.
Yeah, I think it really is something you can discuss with your class, because here two things may happen, right? People may be calling germination different things, so that's one of the issues, and then what happens if you have germination and then that seed dies, right? So you have germination and then the next day you come back for your next data point and it's all dried up. Now, do I still count that as germination? Do I count that as death? And so here, I think it gets to the point of really students are having to define what their experiment is and what they're calling things that they observe in that data collection, because that really plays an important role when you get to the dose response curve. And really, I think this is a great lesson to start those discussions and teach some of those skills to students. And definitely connects into the science and engineering practices that are in the NGSS.
And that definitely goes to the benefit that taking pictures would have. So that's a really good point. So that then after the fact, if students find that their results don't look so similar, then they can really dig deeper into that. So that's a good idea. Thank you for sharing that. And if anyone has any other question or comments, feel free to, again, unmute yourself or put it in the chat or the document.
One thing that I was really curious about was, since I'm less familiar with these ages of kids, sort of what do you think are the boundaries for these activities? Like, do you think it could go all the way down to 6th grade or even maybe like 5th grade, or is that, too young? And for high school, do you think there's a point where this becomes too easy, like maybe 10th grade is the upper limit? Or I know that's kind of a generalization, but maybe you have thoughts on that.
I think you can go either way. I think you can go down to 5th grade, simplifying and having more teacher guidance. I think depending on the 6th graders, in an after-school club setting, we can go a lot of places with this, because you have the teachers there, and we have the older club members and the younger, and we can go places. We have time, and we can pull the 6th graders up to the level of the 7th and 8th graders, and we can also have the 8th graders go farther into the high school. I think your high schoolers might want to push for more interesting chemicals, you know, and it would be interesting if there was some chemicals given to them that you knew ahead of time were going to spark unexpected germination or unexpected growth, because that could be a little, because they're going to probably predict, oh, all this stuff's going to kill it, you know, and then suddenly it's like, wait, no, everything's really, whatever this is, it's doing surprising. You want to kick in surprises and have the kids be hooked by those surprises. Like the plants don't do what they think they're gonna do.
That would be really fun to think of some surprise compounds that have unexpected effects. I like that.
I wonder if there are mixed seed radish packs, because I think there are, right? So
There definitely are. I saw those and avoided them. That would be an interesting twist, yeah.
does it matter what, you know, like I don't know enough about the mixed seed radish packs. Do they have different colors or different styles, types, varieties?
Yeah, I saw some with different colors like white, purple, red, and then they also had slightly different shapes. So yeah, you could do a whole bunch with that.
Yeah, do white, purple, and red all react the same way? I really don't know, but kids they'd make predictions and forecasts, and if we had a little bit of advanced knowledge, we could be like, hey, what do you think? Well, let's try the coffee. For all I know, the purple reacts really well to coffee and the white doesn't.
Yeah, I guess that would lend itself really well to the genetics portion of the discussion. Is that usually, that was part of the middle school NGS, right?
Yeah, I think a lot of the kids have a surprising amount of pseudoscience with cloning and genetics through anime and mangas and many science fiction shows. Marvel Universe.
So thinking about this discussion around, you know, something that maybe surprises students, I wonder what your thoughts are on mixed toxicants versus single toxicants. In this experiment, we talk about just single toxicants and potentially doing a test where you have 2 single toxicants and then mixing those toxicants and running the same experiment to see if you see different results. I don't know if you have any thoughts about that, or if that's something that actually you do, you know, in the real world.
So I actually have a real world connection. I just got back from just before this meeting, Al's Garden Center. I had to pick up fertilizer for the lawn. And so I had a conversation with the Al's Garden guy to, you know, get the soil as neutral as possible. So coffee's very acidic, right? So is there a combination of 2 toxins that will neutralize each other if used on the radish seeds?
Yeah, I think that's a really good thought. And it's definitely a very real world application. I think in our department, there's a ton of people that are really interested in these like complex mixtures of chemicals because they can often behave differently when they're combined together than they would alone. And things get really complex really fast, and we don't really know in a lot of cases how they're going to interact with each other. And so I think that would be a fun direction for the activity if we're looking to up the difficulty or interest level.
I had another question in getting sort of a more of an understanding from a professional toxicology is dose response curves only work for particular toxicants, or there's some toxicants where these dose response curves don't work? And what are your thoughts about explanations of that, you know, as sort of an extension to this lesson?
Yeah, there definitely are chemicals that don't follow the standard dose response curve format, kind of that S-shaped curve. The immediate thing that comes to mind is endocrine disrupting compounds. So chemicals that mess with our endocrine systems, our hormones like estrogen receptor antagonists and things like that. So that I think of a lot in an animal model system. I'm not sure if I can think of a good example for radish seeds. But in those cases, a lot of the time, those endocrine disruptors will have not an S shape, but maybe more like a U shape or the reverse. And those are called non-monotonic dose response curves. So there are different shapes that you could definitely see, and it could definitely stimulate some interesting discussion. Yeah, there are certain areas that as we were creating, we kind of wondered if we oversimplified a little bit, and we did that largely for clarity, the sake of clarity. But there are definitely a lot of nuances that you could dive into more. Yeah, I'll have to think about what might do that for radish seeds, though. If I think of anything, I'll let you know.
Yeah, I was thinking, as an extension, not necessarily with the radish seeds, but just letting students know that there's multiple different ways toxicants work, and it's the understanding of those different ways that help to really create policy or create regulations around what we're using and what we're putting in the environment, and potentially things we don't know about that we're continuing to use. And we hope that our students, our middle school students, our high school students at this point will be solving those problems in the near future. We're getting close to 5 o'clock here. I'm just wondering if there's any final questions. I'll definitely hang around and answer any questions that I can, but I definitely want to thank Team Talks for all their work over the last many months of putting this together. I really encourage everybody to use the activity, and feel free to reach out to me with any comments or questions, and I can get those to the team. We will be having this put on our website along with this presentation. So if you need to check back and get any of the question answers that we did here, you will be able to do that. We'll have all the lessons up. All the resources, they'll all be up on our website. And then I shared the Team Talks website, but there's your YouTube site as well, right?
Yes, that should be at the top of the chat, but I can repost it also. Okay,
yeah, I must have missed that. And I saw that Yvonne just posted the dose response curve generator. That's would be a pretty fun tool that students could use as well.
So I'm thinking about distributing the packs.
Yeah.
The bags that you created this week. Start that process.
So I've talked with teachers who have already done it. High school, mostly high school teachers. You may be the first. I don't know if at least I haven't talked. I don't think I've talked to a middle school teacher now that I think about it that's done it, but a number of high school, and it works very well. Like things just work. And this was done prior to the videos. Since the videos have come out, a couple teachers have done it, and the videos just make it much easier to follow along to do the experiment. To where I think one teacher actually did it completely offline, where the students just watched the videos, set it up, and then they did the discussion with the data around building the dose response curve. So, but that was, again, high school.
We'll probably have a mixture of, because of the COVID reality, we have a mix where we have as club members who are currently in high school and some club members that are in middle school. So it'll be interesting to see how that goes. Yeah,
I'll be interested to hear, and I really think it's a great lesson. the resources and the lesson plan and the support worksheets are really good and have been tested. So hopefully it goes well for you, but definitely let us know.
Sounds good.
Okay.
Thank you so much for your feedback, and I love your background picture.
Oh, thanks. My daughter and her cousin did that, so
It's awesome.
they rather enjoyed Mandalorian.
Okay. Well, thanks for attending, Robert, and we'll see you in another one of these again soon.
All right. Thank you so much, guys, for everything you've done. Appreciate all your work.
Thank
Bye.
you.
Thank
See you
you.
later. Bye.